So in this video, now we are going to discuss about the alternative pathway, which is the part of complement system pathway. So it is the second major pathway which can follow if the classical pathway is not following. So in the condition when will the alternative pathway be activated? Now there are certain bacterial spores, there are certain bacteria, there are cell walls, viruses, fungi, which enter the plasma and blood, but they go unnoticed. Means suppose there is a rarest of condition where they have not bound to IgM or IgG. In that condition, C1 will not be activated. Now please remember that if this is your C1 protein and if this is your particular antibody, so here on the C1 protein we have Fc receptor. This Fc receptor is the one which is holding this antibody, so you have to remember this. Okay, now suppose there is a condition where the bacteria, its cell wall, or a virus, any spore or fungal spore, it enters the blood but it is unnoticed, the antibody has not bound to it and C1 is not activated. In that condition also the complement system will tackle it because of C3. Now C3 here is a main hero. On the C3 there are certain thioester bonds. These thioester bonds are very unstable, means they degrade naturally, they undergo hydrolysis. This hydrolysis was not happening in the classical pathway, but here it is happening. So C3 generally and randomly undergoes hydrolysis to form C3A and then C3B. C3B. This will be removed, this will come. Now it will bind to a particular such C3B molecules, they can bind to a particular cell which has low sialic acid, SIALIC, sialic acid secretion. If low sialic acid secretion is there, then these randomly synthesized C3B molecules will be binding to it. Now here is the point of understanding. All the mammalian cells, they have high concentration of sialic acid every time. So even if the C3B molecules are being produced randomly, they will not bind to these mammalian cells because the mammalian cells are secreting high concentration of sialic acid. But those bacteria, cell wall there, and the virus and fungi, they do not secrete that much amount of sialic acid. In that condition, these randomly synthesized C3B molecules start binding to the bacteria. Now the C3B molecules, once they start binding to the bacteria, they play a very important role there because they are now activating the alternative pathway. So here what happens, C3 randomly fragments into C3A and C3B. Now this C3B is very important because there are certain other factors, other complement proteins. One of the such complement protein is factor B, one of them is factor D, and then one of them is properdin. There are three of them. So let us see how it happens from here. So this is a C3 molecule. So let me change the color. So here is a C3 molecule. This C3 molecule will be randomly fragmented into C3B and C3A. This C3B under low sialic acid concentration will bind to a particular cell. Suppose this is that bacterial cell and here the C3B molecule is attached. Now onto this attached C3B molecule, the factor B will come and attach. So factor B will come and will attach to this. Now the factor B under the presence of factor D, factor B under the presence of factor D breaks into BA and BB. This BA again leaves and the BB stays. So here this is your the bacteria, this one is your C3B and this is your B small b, the B part of the B factor. Factor D has no important role in joining, it is only there to cleave this B. So B is cleaved into BA and BB in the presence of factor D. Now this particular complex of this two proteins, it is very unstable. It has half-life of only 5 minutes. Now to stabilize it, we require another important factor, that is properdin. So properdin is actually trying to stabilize this particular complex. Now this particular complex, which is C3BBB, acts as C5 convertase. It will then break the C5. So C5 under C3b BB will break into C5A and C5B. This C5A, as I was telling in the previous video, is a major inflammatory agent, and this is what we call as such inflammatory agent. C5A specifically is called as anaphylatoxin, which causes the inflammation. And the C5B, again in the presence of 6, 7, 8, 9, all of them together, they will form the membrane attacking complex. So the remaining part is very simple. Please remember that when I talk about anaphylatoxin, it means that these C5A molecules will go to mast cells and they will cause its degranulation. Means the granules will undergo exocytosis to release their components. The components include prostaglandins, prostaglandins, leukotrienes, and bradykinins. The three of them are released. These two will be means the three of them, not two, the three of them will be responsible for inflammation. So this is the major function of C5A, which acts as anaphylatoxin. So this is the alternative pathway of the complement system. Now we will in the next video talk about the third important pathway of complement system, that is mannose-lectin binding pathway.